title of the award or the grant is Assessing Long-Term Stability of Cerebrospinal Fluid PRP Levels in Genetic Prion Disease Mutation Carriers. But really what this is about is how do we prepare to test whether a treatment is actually going to be helpful for prevention and treatment. And so that really is the program that this award is helping to fund. And I first want to acknowledge Eric and Sonia, really who inspired this work and have been basically the co-drivers of the work. They are now going to be joining us at Mass General Hospital as well as the Broad Institute to really help develop the program even further. These are some of the folks that are involved in the study. Chase Wennick is our clinical research coordinator. Allison McManus is our nurse practitioner, who is now doing a number of the spinal fluid tests. Tori Williams is our neuropsychologist. Becky Carlisle runs the lab, and we have other folks as well. And I also want to, of course, acknowledge and especially thank the family grants and funds that have supported this work through the Creutzfeldt-Jakob Disease Foundation and also with some initial funding from the Prion Alliance. So I'm not going to belabor this. We're all here because of this. We have Creutzfeldt-Jakob disease, we have GSS, we have fatal familial insomnia. These are the different flavors of one really common disease, which is based on prion pathology. And as we know, and as we've seen in other slides yesterday, we all make prion proteins. If you have a mutation, you may be more vulnerable to having your prion proteins misfold in a way that actually serves as a template to take normal prion protein and convert it into the abnormal, bad, toxic, and basically indestructible pathogen that it is. And this goes through a chain reaction of misfolding and spread, and ultimately leads to what is called a spongiform encephalopathy, as we've seen. And this is a picture of under a microscope of this spongiform encephalopathy, where you've gotten massive death and dropout of neurons, leading to the progressive neurodegenerative dementia that this is. The rationale, as Dr. Kai just showed in the video, one of the rationales for one of the major treatment approaches we have is this ASO, or antisense oligonucleotide. So I'll just very, very briefly talk about what this is. So we all have genes. We all have DNA. DNA makes messenger RNA that floats out into the cell body and serves as a template to make the proteins, which then normally fold normally. And then, as I just described, these normally folded proteins can sometimes misfold and cause the pathology that the disease is. What ASOs do is you can synthesize a little bit of RNA here, which will bind to this messenger RNA, and then lock it up, and then you put something else in, and it causes the RNA to degrade. The net result of that is you don't make the protein, and then if you don't make the normal protein, you don't have any opportunity to make the abnormal protein. And that is the rationale behind this ASO therapy. So the question is, we can develop this, we can show that it works in mice or in other animal models. How are we going to know whether it actually works in a human? So there are a variety of ways in which we do this for treatments, particularly for preventive treatments. One, We could give someone the medicine and then just wait to see whether they develop the disease. That could be 2 years from now, 20 years from now. We can look for early signs of disease in terms of cognition, disease progression, survival, like we do with the animal models. But the major way in which we're going to be able to determine whether an ASO works is with this thing called biomarkers. And biomarkers have different forms. You can have, basically what a biomarker is some biological measurement, whether it be an X-ray test, an MRI scan, a blood test, a spinal fluid test, an EEG test, any of these things that can tell us how the brain is functioning even before there's a disease that's going on. So the program and the platform that we have developed is really to start looking at these biomarkers with a particular emphasis on spinal fluid, and I'll show you why. So far, we put the call out, and mostly by word of mouth and through the CJD Foundation and the Prion Alliance. We have had 43 people come into the study so far. The rationale, research participants will call Chase Wennick, our research coordinator. He'll do a little screening on the phone. He'll tell about the study that we're running. What we're trying to do is have people come in for an evaluation, come back 8 weeks later for another evaluation, because we want to see that what we measure is actually stable. And I'll tell you a little bit more about why that's important. So we've had 43 people so far. 27 are known carriers of a mutation for prion disease. 11 are known non-carriers in families, and or family members without a family history, meaning spouses and the like. We have a few that are still, we're waiting on the genotypes. This is the age distribution, the sex distribution. They're generally a pretty highly educated groups. About 45% have some of the carriers have some family history of CJD, 30% FFI, and 25% GSS so far. And in terms of the clinical characteristics, we do some physical examination, neurologic examination. We do some health questionnaire screening. We do some cognitive testing. We do some neuropsychiatric scales of depression and anxiety. And by and large, people, whether you're a carrier or a non-carrier, people look really pretty normal. There's a little bit more anxiety and depression among the carriers, but not anything that we would consider to be abnormal or a mental illness. So overall, people are looking pretty similar, whether you have the mutation, and I think that's what we all know. In our family members who may have developed the disease, they were completely normal until this thing took over. So there are a number of biomarkers that are used. Right now, we are focusing on EEG. We're collecting blood and spinal fluid. With greater funding, we would probably like to do some MRI work or even PET scan work, but we're not doing that right now. The EEG is a way to measure brain waves, and we're looking to see whether there are any subtle abnormalities. And we've got Hamed Azami, who's our bioengineer, does signal processing, is doing some very fancy kind of artificial intelligence type of analyses of the EEG signals. So far, we're not seeing any real abnormalities. The EEG in carriers of prion mutations looks pretty much normal. And again, I think that reflects how normal their brain is functioning in general at this stage. And our goal is to keep it that way. But spinal fluid is where the real action is. And because spinal fluid is a fluid that is produced in the brain, bathes the brain, and is accessible clinically, you can get a tremendous amount of information about the molecular status and the molecular milieu of the brain. So I'll show you, so this, just to kind of demystify this whole process, this is me actually with one of our early participants. You know, it's been called lumbar puncture or spinal tap. Those have bad connotation. I'd actually like to shift the term and just say spinal fluid test, like blood test. But spinal fluid is kind of made in the deep cavities of the brain. It kind of flows out and around and up and down the spine. And you can see it in blue in this. And the spinal cord goes down to about here. And then there's just this sac, this empty sac. Well, there's some nerve roots that float around in there. But the sac of fluid down here. And then in a actually relatively simple procedure, you can use a very skinny needle. It's even skinnier than what's used in blood tests in order to sample the fluid from there. You roll up your sleeve for a blood test. You roll up your shirt for a spinal fluid test. You know, we do it under sterile conditions, so we wipe the skin down with iodine soap and we give a little numbing medicine under the skin. But the whole thing usually takes about 15 minutes or so, and then you go about your business. Doesn't cause paralysis or any of the things that people are always afraid of. The reason why we're doing this is because if you have an ASO that can knock down the amount of prion protein that's going to convert into the bad prion, we want to know, can we measure it in spinal fluid? And then if you give the drug, will it knock it down? Okay, so how can we know whether we do that? What if the prion levels are kind of fluctuating day to day, you know, and then you've got, you know, you do your spinal fluid test, and you know, and then the next day the prion level is up or down, and it just normally fluctuates. So we really needed to see, is this a stable measure? Can we use this as a biomarker outcome of target engagement for the ASO therapy? And so people came back you know, they had the baseline spinal fluid, then they came back 8 weeks later, and in people with various mutations, and now we're moving into annual follow-up. We are finding that within any given individual, the prion levels stay stable. Okay, this is a good thing because it shows that our target is stable and that if we can give the drug and knock it down, then it recapitulates what we want to see and what we do see in the animal models. And this is really a critical thing. And the FDA, where we had a meeting, Eric and Sonia and others and I went down to the FDA, and they are recognizing that this is a valid outcome. So, okay. So we're able to measure many, many other things in spinal fluid, and that's why it's so valuable. We can measure tau, we can measure neurofilament, we can measure inflammatory markers. So there's, you know, aside from just looking at the prion levels, there's a tremendous amount of information that we get about the health of the brain that you can't get from any other biomarker that we have. And finally, we're asking folks what their experience is when they get the spinal fluid test, because it does have such a stigma. So we give them a little scale about how anxious they were. We give this before they get the spinal fluid test, after they get the spinal fluid. How anxious are you? In anticipating, how anxious were you, and how painful was the procedure. And as you can see, by and large, most everyone is a little bit anxious when they start, and then afterwards they say that they're much less anxious, that it wasn't painful, and that they're not anxious about getting the next spinal fluid test. So that's the experience that our folks are having in this. So again, I'd just like to acknowledge our team, our funders, and if you're interested in participating, this is the contact information for Chase, and then also my email as well. Thank you.